Why do 100% of scientists geek out over DNA shapes? Let's dive in. DNA isn't just a boring double helix, it has three cool forms, A, B, and Z. Form A is like DNA on a diet. It's short and fat. Structure, helical orientation, right-handed helix. Pitch and diameter. The helix completes a turn every 2.6 nanometers, 26A, and has a diameter of about 2.3 nanometers, 23A. Major and minor grooves. The major groove is narrower and deeper compared to bDNA, while the minor groove is wider and shallower. Base pair orientation. Bases are tilted about 20 degrees relative to the helix axis. Stability, formed in dehydrated conditions or in the presence of certain chemicals. Less stable compared to bDNA. Biological relevance. aDNA is typically found in DNA-RNA hybrids and in some DNA-protein complexes. It is important for understanding DNA conformational flexibility and interactions in various biochemical contexts. Found in dehydrated conditions, it's more compact and less twisted. Picture a squished spring. Form B is the classic spiral staircase you see in textbooks. It's the most common form in our cells, flexible and perfectly balanced. Helical orientation, right-handed helix. Pitch and diameter. The helix makes a complete turn every 3.4 nanometers, 34A, and has a diameter of approximately 2.0 nanometers, 20A. Major and minor grooves. Prominent major and minor grooves, which are crucial for protein-DNA interactions. Base pair orientation. Bases are perpendicular to the helix axis, contributing to a smooth, uniform helical shape. Stability. Most stable under physiological conditions due to its high hydration level. Biological relevance. The B form is predominant in vivo and is essential for DNA replication and transcription. It serves as the foundational model for understanding DNA's biochemical interactions and processes. Now, Form Z is the rebel. It twists the opposite way in a zigzag pattern. This form is rare and pops up under stress. Structure. Helical orientation, left-handed helix, pitch and diameter. The helix turns every 4.6 nanometers, 46A, and has a diameter of about 1.8 nanometers, 18A. Major and minor grooves. The major groove is almost absent, while the minor groove is narrow and deep. Base pair orientation. Bases adopt a zigzag conformation relative to the helix axis. Stability. Stabilized under high salt conditions or specific DNA sequences such as alternating purine-pyrimidine repeats. Biological relevance. zDNA is associated with gene regulation and chromatin organization. It can play a role in the formation of certain genetic elements and responses to environmental stresses. Think of it as DNA's way of saying, I need a break. So, why do these forms matter? They influence how DNA interacts with proteins and how genes are expressed. Understanding them can unlock new medical breakthroughs. And there you have it. A, B, Z, the three funky forms of DNA. Smash that like button if you learned something new.